non-disjunction. Disjunction is a process by which homologous chromosomes they separate from each other. This is a process in case of mitosis where the sister chromatids they separate from each other. While on the other hand, if the homologous chromosome or the sister chromatids they fail to segregate from each other, such a phenomena is called as non-disjunction. In this diagram, we can see that chromosomes are arranged at the metaphase stage, that is in the middle. These are the chromosomes in the form of homologous or chromosome. Two sister chromatids are joined at the position of the centromere. In case of proper disjunction, we can see that the chromosome is divided into two sister chromatid, one sister chromatid and second sister chromatid, and each of the sister chromatid will move towards the pole, one pole and the second pole. While on the lower hand, if we see that both the sister chromatids they are moving toward one direction. So if there is a proper disjunction between the sister chromatids or between the homologous chromosome, that is called as disjunction. And if there is non-disjunction, then the separation between the homologous chromosome fails. There are two types of the non-disjunction. First one is mitotic non-disjunction and second one is meiotic non-disjunction. In case of meiotic non-disjunction, if the disjunction is during the prophase of meiosis 1, then all the sperms or all the ovas which will be produced from the primary spermatocyte or from the primary oocyte, they will be abnormal. Failure of the separation of the chromosomes can be of the three types. This failure can be during the first meiotic phase where the homologous chromosome they separate from each other. It can be during the meiosis 2 where the sister chromatids they separate from each other. Failure of the sister chromatids can be during the mitosis. In case of mitosis, sister chromatids they fail to segregate from each other. Meiotic disjunction produces two cells. These two cells they have different number of chromosomes. Different number of chromosomes is due to non-disjunction of the centromeres of the homologous chromosome. They will not separate from each other. If the spindle fibers they are attached to the sister chromatids, they do not attach to one of the sister chromatid, ultimately they will not separate from each other. One of the cell will contain both the chromosomes or both the sister chromatids, while the other will be lacking of the chromosome. Mitotic non-disjunction produces two types of the cells. One of the cell will be trisomic in nature, and while the other cell will be monosomic in nature. If we see in this diagram to understand that what is non-disjunction and normal disjunction. This is a cell with 46 chromosomes. Consider this cell that this cell has 46 chromosomes initially. As a process of non-disjunction, if there is non-disjunction, then one of the cell will have both the chromosomes, while the other cell will be lacking the chromosomes. As a result of the second meiosis, the cells will be produced with 24 chromosomes and these cells will be with 22 chromosomes. So if the cell has 46 chromosomes, then as a result of meiosis, the cells they should contain 23 chromosomes. So if we see, if the non-disjunction is during the meiosis 1, then all the cells which will be produced, they will have abnormal number of chromosomes. While on the other hand, if we see that the first meiosis is the normal one, where the chromosomes they segregate, one of the chromosome is in this cell and the second chromosome is here, because the first meiosis is the normal one, and if there is non-disjunction during the meiosis 2, in this case the cell will have 23 chromosomes, while because there is non-disjunction during this meiosis 2, this cell has 24 chromosome while this cell has 22 chromosome. So we can conclude that non-disjunction can be during the meiosis 1 and it can be during the meiosis 2. If it is during the meiosis 1, then all the cells which will be produced will be abnormal. While in case of meiosis 2, because the chromosomes they have been segregated properly during the meiosis 1, so if there is non-disjunction during the meiosis 2, some of the cells they will have normal number of chromosomes, while some of the cells they will have abnormal number of chromosomes. In this diagram we can see that as a result of non-disjunction, if non-disjunction is during the first phase, then the cells will be produced, they will be trisomic and monosomic. So two of the cells they are trisomic, while the remaining two cells they are monosomic. While on the other hand, if we see, if the second division has non-disjunction, then these cells they are the normal one, while these two cells they are either the monosomic or the trisomic. Causes of non-disjunction. There are several causes of non-disjunction. The important causes are the age of the maternal, mean the age of the female, the radiations which are present in the environment, as well as delayed fertilization after ovulation.